Hello, good morning, children. My name is Sujata Savarkar, and I'm your biology teacher for grade 7. Children, we are all going to have very nice fun once we are back to the school with lot of activities coming up for the subject, okay? Because biology is all about the nature, the study of the plants, the animals, and the nature around us. So, for to start with or to begin with, we are going to start with the lesson tissues, okay? Now, this lesson tissues is composed of two parts. One is plant tissues and animal tissues. But for today, let us simply focus on plant tissues. We are having one PPT which is going to help us to study plant tissues. So let us study PPT. So children, let us begin with the PPT which is based on the lesson tissues. The learning objectives, obviously we are going to have the concepts of plant tissues, the types, the locations, the structures and the functions. Also animal tissues, their types, the location, structures and the functions. Today's learning, for today we are going to have plant tissues, what are its types, the structure, the location, all the parameters. Have a look at this flow chart over here. We can see plant tissues being bifurcated into two, meristematic and permanent. Meristematic cannot, they keep on dividing in fact, whereas permanent, they stop dividing. Meristematic further divided into apical, intercalary and lateral. Permanent divided into simple, complex, and furthermore divided into parenchyma, collenchyma, sclerenchyma, and complex being divided into xylem and phloem. So you'll all practice this flow chart in your practice other of notebook. Okay, it's very important. To begin with, let us start with the meristematic tissues, the first kind. So these are the general features of the meristematic tissues. These are living cells. They are very closely packed with no intercellular spaces. The cells are very similar in shape and they have got thick protoplasm, thin cell wall because they have to keep on dividing. Okay, and this is also referred as the primary growth. The meristematic tissues are of three types: apical, intercalary, and lateral. You can see apical at the top, intercalary in between, lateral on the sides. What are they? Let us study them in detail. Apical meristem, these are found at the tip of shoots and roots. So they obviously they are going to increase the height of the plant, and therefore this is called as a primary growth. The apical growth is called as the primary growth. Lateral meristem, obviously the parallel sides. So they bring about the increase in the girth, or you can say the diameter or the thickness of the stem. So you can see the cork and the vascular cambium. Here is the growth happening, and that is why they bring about the increase in the thickness. This is called as the secondary growth. Lateral meristem. Okay, next is intercalary. These are present at the base of the monocots, at the nodes, internodes, and the leaf base, and therefore they bring about the growth at these regions. That was all about meristematic. Let us go forward towards the permanent tissues. The meristematic cells, once they mature to form the permanent tissues. Then they stop dividing and they acquire a very definite shape, form, and size. So these cells could be living, dead, or and nucleus at the borders. See, focus on the permanent tissues here. Simple, complex. Simple into parenchyma, collenchyma, sclerenchyma. Complex into xylem and phloem. So let us study what are simple tissues first. So these are made up of dermal tissues and the ground tissues. Simple tissues divided into dermal and ground tissues. Dermal tissues. These are very closely packed. Dermal are just similar to our skin. Okay, they cover up the organs or the organisms. So their function is to protect. Outermost layer is called as the epidermis, and obviously it has got some cases waxy coating to retain the water and the leaves don't dry. Epidermis also sometimes has stomata for photosynthesis and gaseous exchange. Okay, yeah. Next is ground tissues. After simple tissues, ground tissues. There are three types. You can see had this the type of cells: parenchyma, collenchyma, and sclerenchyma. But to begin with, parenchyma. These are thin-walled, large vacuoled because they have to often go cell division. There is no intercellular space. Their function is to prepare the food and store the food. And in case they have got chloroplast, it is called as chlorenchyma. And when they have got air cavities, they are called as aerenchyma. Exam point of view, please study this very properly. This is very important. The next is collenchyma. You can see they are thick only at the corners because of a substance called pectin, and they will be present at the or below the epidermis of dicots. It obviously provides mechanical strength and they give elasticity and flexibility. If you see a plant is able to move on its fixed place in case of a wind, that is because of collenchyma. Sclerenchyma, these are very thick walled, okay, these are very thick walled because of a substance called as lignin. Their again job is to give mechanical strength, and there are two types, sclerites and fibers. Sclerites, these are thick and lignified with pits. These are found in seed coats and pulp of fruits. It gives textures and crunchiness. Whereas fibers, there are pointed ends found in the midrib of the leaves and xylem and phloem. So this is now complex tissues. Complex tissues, we saw that it is made up of xylem and phloem. This is an overview of based on parameters, how are they different from each other. Okay, but individually to study, let us see what is xylem. It is made up of mainly dead cells. They are elongated and arranged end to end, forming a network of tube-like structures. They have thick cell walls composed of lignin. Pits are present at the all over the walls. Okay, and tracheids and vessels are the main elements. You can see tracheid, vessel, parenchyma and xylem fiber. Out of it, tracheids and vessels are the main elements. So tracheids have tapering end walls, whereas vessels the end walls break down so that they form a pipe-like structure, and the main function of the xylem is to conduct water and dissolve minerals upwards from the roots to the other parts of the plant. Whereas phloem, it is made up of living cells and elongated, and they are tubular again. Sieve tubes and companion cells form the main component or the elements, and their main function is to transport the food from which is prepared in the leaves to the other all the parts of the plant. So here it will be bidirectional, whereas xylem, water will only travel from the roots to the other parts, that is upwards. And to note that these have got the sieve plates which have got sieve pores, so that food can transport from one tube to the other tube. See this, so that the sieve plate have got sieve pores which can enable the food to transport from one part, one department, one tube to the other tube. This is comparison between xylem and phloem. Please have a look at this and also study and learn the table of differentiate between given in the textbook or the PDF for now. Okay. Now go through the and learn the table given. That is what I have told you again. And please read the fact file as also. That can also come in the paper. And for last but not the least, there is a Google test prepared for you. Based on today's content. All the best, children. I hope you have understood what we have learned today. Go through the PDF properly, and see you very soon for the animal tissues. Bye bye. Have a nice day, children.